Welcome, everyone. My name is Susan Law. I'm the Knowledge Mobilization Lead for Long COVID Web and delighted to have you all here today. And also delighted to welcome Dr. Michael Peluso and hopefully Dr. Danny Altman. We haven't seen him yet, but we're hoping that he's able to join us for this session. And if not, we can reschedule him. And Dr. Peluso has agreed to carry on with extending his presentation. So we'll play it a bit by ear on that one. But I just have a few bits of order of business here to start with. I'd like to start with a land acknowledgement. We wish to acknowledge this land on which the Long COVID Web operates. For thousands of years, it has been the traditional land on the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. Today, this meeting place is still the home to many Indigenous people from across Turtle Island, and we're grateful to have the opportunity to work on this land. A reminder of the purpose of these webinars. First, to share evidence-based information, research results relevant for decision makers, researchers, clinicians, patients and families, and the general public. Secondly, to provide ongoing updates and spotlights on emerging evidence, as well as scientific information about the activities of our national network. Thirdly, to contribute to reducing and managing misinformation about long COVID. We do have a code of conduct, and the link is there in this slide, and it's also online. We value highly engagement around the science and its interpretation as well as its application. A commitment to reasoned discourse, as stated in our code of conduct, comes with the responsibility for all to ensure that any comments or questions remain within the spirit of positive, constructive, and respectful discussion. So we welcome any and all questions and comments in that spirit. Thank you. Your cameras and mics are off, but please post your questions in the chat. We will pause for the Q&A after the speaker and speakers, we hope. We will do our best to review and address as many questions as we can. And these sessions are recorded. So with permission from the speakers, they will be posted online following the webinar. And we do welcome your thoughts about how these sessions could be most helpful to you. We have a very brief survey. Please take 2 minutes to fill it out and give us some of your ideas about what else we could be doing with this time together. Thank you. So there's the link to our survey. And I'm just going to stop sharing my slides and turn it over to Dr. Michael Peluso, who is an infectious disease clinician and a researcher, associate professor of medicine at the University of California in San Francisco. So thanks, Dr. Peluso, for joining us today, and over to you.
Hey, everybody. It is a pleasure to be here to talk with you today. So I'm an ID physician and researcher at UCSF. My initial plan in my research career had been to focus entirely on HIV and HIV cure. And then almost 6 years ago now, when the COVID pandemic began, I worked with a big group of people at UCSF to kind of pivot our research program to understanding the consequences of COVID over weeks and months and years, sort of on an informed, informed hypothesis that this was probably going to be more complicated than how it was being described at the time. 2 weeks of illness and then full recovery. And so unfortunately that turned out to be the case. And really, since that time, my major focus has been on long COVID and really understanding biologically what might be driving long COVID. So I'll talk today mainly about our work over the last few years in viral persistence and immune dysfunction. And then depending on time, I'll talk about some of our upcoming clinical trials at UCSF in the US, and then some other programs that we are working on or that I think we should be working on. And we'll sort of play it by ear with regard to the timing of things. Here are some disclosures. I want to begin by acknowledging our research team at UCSF. It's actually, as many of you know, this is quite a complicated topic to study. The level of complexity is really high, and it takes a lot of people to do it right. And so it's really been a pleasure over the last few years to get to know um, everyone on our team and to sort of see them develop and gain skills in this area. And I think for our team, it's really a privilege to work with patients with long COVID, try to figure out what's going on and get them better. So our program in San Francisco is called LINC, which stands for Long-Term Impact of Infection with Novel Coronavirus. And as I mentioned, we opened in April of 2020 to understand what would happen to people really immunologically after they had COVID. Within a couple of weeks of opening, we began to encounter people with what now would be considered long COVID. We realized that that was, seemed really clinically important, and that's been our focus ever since. Since that time, we've enrolled over 1,700 people. We've conducted thousands of study visits. We've banked a lot of biological specimens, which we share across multiple collaborations. Have had dozens of collaborations across the US, North America, and around the world. And then, within LINC, we have an acute COVID program where we enroll people during a new COVID infection and follow them prospectively to look at the biological determinants of long COVID. We have a tissue program where we're doing gut biopsies, lymph node biopsies, lumbar punctures, bone marrow biopsies, and endometrial biopsies to really understand what's going on in the tissues of people with long COVID. We have an imaging program where we do PET imaging with sort of research, non-clinical tracers to understand what's going on on a whole body level in terms of inflammation. We have a clinical trials program that we're very proud of. And then recently we've begun expanding our infrastructure to other infection-driven chronic illnesses, including ME/CFS. And then just to say, since 2022, we've also been part of the US national program, which is called Recover. And so within UCSF, we have both kind of our local program and also we contribute to the national program. The thing that we're most proud of is how far our biospecimens go to really support research that's happening to understand what's going on in people with long COVID. And this is just a snapshot of investigators and collaborators that we've shared LINC biospecimens with. And you can see they're quite a diverse group of people all across the world. This is from a paper that we published a couple of years ago, sort of laying out our conceptual framework for long COVID. And the way that we think about this is that obviously, a person has to have had acute COVID. Then there's kind of the upstream biology, abnormal biology that that COVID infection causes, which are things that we really measure in the research lab. So processes like viral persistence, immune dysfunction, autoimmunity, mitochondrial dysfunction. And then that biology drives what we refer to as abnormal pathophysiology, which are things that we measure in the clinic. So organ system dysfunction, dysfunction of various regulatory axes, dysautonomia, cognitive dysfunction. And ultimately that abnormal physiology is what is experienced by a person as long COVID. And within LINC, we have programs that are focused on all of the different aspects of this, some of which are quite mature and some of which are in the earliest stages of development. But we're kind of going down all of these different pathways. So the pathway that has been our biggest focus over the last few years, not because we're married to it in any way, but because we've had some funding to study it, was SARS-CoV-2 persistence. And I'll say up front that, I was actually not really a believer in this at the beginning of the pandemic, but my thinking has evolved as the data has emerged. So this is a really beautiful review that was published a few years ago by Amy Proal from Polybio, which is a foundation that supports work on long COVID, where she cataloged all of the different studies that went against kind of the textbook framework that SARS-CoV-2 by definition has to be a transient virus. Um, all of these studies have major shortcomings, including the ones by us. um, but the message here is that there are dozens of studies by different groups working independently in different cohorts using different assays that are ultimately coming to a similar conclusion, which is that at least components of this virus stick around for longer than we would expect in at least some people. Like I said, all of these studies have major limitations, so this is not definitive. But I think it's not really controversial anymore that this is happening in at least some people. I think a problem that I see is that this has become really polarizing, and people are either, 100% this must be happening and this is the cause of all disease, or that this is like made up and impossible to be happening and we shouldn't even study it. And that sort of very black and white view of things, I think, is a big problem for the field that I'm trying to, address. My position on this is I don't care whether it's happening or not. We just need to know, and then we need to deal with it in either way. So, what convinced me of this was an initial study that we did with David Walt's group at Harvard, where we shared hundreds of biospecimens from both before the pandemic and in people post-COVID for measurement using a single molecule array assay, which is basically a research-only test that measures really minuscule amounts of proteins. And so we shared blood samples, and what we found was that in the pre-pandemic samples collected, before anybody on the planet had COVID, about 2% of them were positive on this test. Those are false positives. It's impossible for those people to have COVID. It's cross-reaction. It's not real. But that means that the specificity of the assay is 98%, which is not bad. That's pretty good. But when we looked at people post-COVID, this is all comers, regardless of whether they had long COVID, we found that there was a significantly higher proportion of people with detection of these antigens circulating in their blood 3 to 6 months post-COVID, 6 to 10 months post-COVID, 10 to 14 months post-COVID. You might actually convince yourself that it kind of wanes over time. We've now extended this out to about 2 years, a little beyond 2 years, and we see sort of a similar pattern of detection. And so, what this means is that we're detecting something that we think is related to the virus, is a component of the virus, and that we don't think should be there, and that it's happening at a rate that's higher than in the background of what we could see before anybody had COVID. In addition, not shown on the slide, we actually saw in this publication that there was a significant relationship between how sick a person was during their initial infection and how likely they were to have this, months after they had that infection. So people who had been hospitalized with COVID were about 3 times more likely to have this compared to people who were not hospitalized. And even among people who were not hospitalized, those who just said, I was really, really sick at home versus, oh, it was pretty mild, were, again, 3 to 4 times as likely to have this. And so as a clinician, to me, that's really compelling that there probably is something truly biological going on with this. A big focus of our more recent work has been trying to tie this specifically to long COVID. And the data are not quite as clear-cut there. This was some data that we presented at one of the infectious disease meetings last year, where we looked at the relationship between detection of circulating spike protein in blood 3 to 14 months post-COVID and how likely a person was to meet criteria for long COVID. And what we did was we specifically looked at different categories of long COVID to address some of that variability between patients that can sometimes be a challenge. And so what we saw is really a signal that we thought was emerging between detection of circulating proteins in blood and neurologic long COVID symptoms, cardiopulmonary long COVID symptoms, interestingly, musculoskeletal long COVID symptoms. Some of these were statistically significant. Some of these were almost statistically significant. We didn't really see as convincing a relationship with symptoms like fatigue, and that sort of makes sense to me because I think that fatigue is probably the least specific long COVID symptom. It's very common, but fatigue can be caused by so many other things. Um, so anyway, I don't think this is 100% convincing, but it is a little bit striking, and we're trying to build this out further now. A question that often comes up is, well, how do you adjust these models for all of the other things that might drive either these symptoms or persistence of a virus? And I mean, I will say we have spent a lot of time trying to come up with these models where we can adjust for all of these factors that we think are important, and these models are adjusted for these various things. So anyway, more to come on this, but we think that we are seeing something emerging there. There's also, I think, very compelling evidence that is more indirect, that the virus can persist in some people. So this was a project that we did a couple years ago with Nadia Roan at the Gladstone, which is down the street here in San Francisco. And what we did is we looked at people who had consistent post-COVID symptomatology, either people who over a period of 8 to 12 months, you know, were clearly fully recovered from their infection, not even sort of a smidge of any symptom emerging. And we compared those people to individuals who had long COVID and consistently met the criteria for long COVID over that 8 to 12-month period. So what that means is that we excluded people who had kind of varying symptomatology in this study. So we had a group that was really consistently symptomatic over time and a group that was really consistently fine. And then we measured using these really high resolution, very complex immunologic measures, the responses of their T cells, CD4 T cells and CD8 T cells. Um, and what we found was that the group that had long COVID compared to the group that fully recovered had higher expression of markers of T cell exhaustion, specifically on CD8 T cells. And that was really on SARS-CoV-2 specific CD8 T cells, not just like any CD8 T cell. Other groups have found this. Akiko Iwasaki's group at Yale has made similar findings, or some groups in the UK that have made similar findings. So CD8 T cells, which are, the T cells that we think respond to infections, are running out of steam. They're kind of exhausted. And then we also found increased expression of markers on CD4 T cells that suggest that those T cells are actively trafficking into tissue spaces. So even though this is a blood measurement, it suggests that those T cells are moving in and out of the tissues where we would think that SARS-CoV-2 would be persisting. And so a way to put all of this together is that there is viral persistence in the tissues that is causing an immune response that may or may not be counterproductive. This is a beautiful comment that came out in Nature a few years ago, arguing just this, that tissues, not blood, are where the immune system is most active and where we really need to be understanding the activity of the immune system. And so because of these findings, we've put a lot of energy and resources into understanding the tissue biology of long COVID, so looking beyond blood. This began with our tissue imaging program, which is led by my colleague Tim Henrich. This program was actually directly an extension of our HIV imaging program. And so what we do in this program is we do PET scans, but these are not, standard PET scans that you could get at, any hospital. They're research-only PET scans using specific tracers that are not clinically available and that have been really developed to understand the immune system in these tissues. And so we use a tracer, we use the tracer in our initial studies called FRAG. And the way that this tracer works is that it basically lights up activated T cells. It's very specific for T cells. And so if you see signal, you know that the T cells in that tissue are activated. We study people with long COVID. We study people who have fully recovered from COVID, and we also have imaging archive of pre-COVID controls who were studied using the same imaging parameters. And we look across a spectrum of time from infection. And so what we get are these images where then you can sort of delve into individual tissue spaces and understand how bright those different tissues are, which is a direct correlate of how much T cell activity there is. So the first thing that we found when we did this was that there was increased T cell activation in people with COVID, regardless of whether they had long COVID, compared to pre-COVID images. So that means that, well, I actually don't know what that means, but I don't like that. That suggests that the experience of having COVID has basically caused a new immunologic set point for all of us, regardless of whether we've recovered or not. I don't know what that means. I don't know what that means in terms of overall inflammation over years or decades, and whether we will see other consequences of this, but we were quite perturbed when we observed that. We also see that some things really stand out among people with long COVID compared to people who have fully recovered from COVID. And that includes increased signal in tissues like certain neurologic tissues, especially the spinal cord, vascular tissues like the aorta and pulmonary arteries, lymphoid tissues like the nasal turbinates, the tonsils, sometimes lymph nodes, the gut wall, and the bone marrow. And this is not in everyone, but when we see it, it is often quite striking. These are not necessarily the tissues that correlate with specific symptoms that people are having. So it's not as simple as a person has neurologic symptoms and their brain lights up. What we see, for example, what's shown here is that we can see increased activity in the GI tract of people who have cardiopulmonary symptoms like shortness of breath or cough or exercise intolerance. These aren't necessarily people who have GI symptoms, but it suggests that there's something going on in the tissues on a total body level, perhaps regardless of what specific symptoms a person is having. So, we initially saw these changes on non-invasive imaging, and then we thought, well, we need to be looking at what's actually going on, in the actual tissues. And so that was the point at which we launched our tissue biopsy program. And initially, the biopsy program was only in people with long COVID and was really focused on the GI tract because that, is an easily accessible tissue that, is straightforward to get. People understand what a colonoscopy is. Many people have had a colonoscopy before. What we actually do for this are flexible sigmoidoscopies, so they're less invasive than even a colonoscopy. It doesn't require the full prep. And our gastroenterologist, Ma Sumsuk, goes in with the scope and collects a dozen or so different biopsies from the rectal area, and then those samples are processed in a very specific way, go to Tim's lab. And what I'm showing here is RNA scope data, where you can actually see, the gut tissue in blue. The circles are basically the epithelium, and there's this layer of blue, which are the connective tissue layers between the epithelial cells. And you can see in green is what we believe is SARS-CoV-2 by RNA scope that is present in some of these cells. Sometimes it's present kind of adjacent to the epithelium, but often we see it deeper in the tissue in the lamina propria, which is a connective tissue layer where there's a lot of trafficking of immune cells. We actually see it often in or adjacent to cells that we think are macrophages. And on the right of the slide, what you're looking at is in pink, actually double-stranded RNA, which should really only be present if the virus is moving through the life cycle. So that could mean that there is active viral replication, or it can mean that there is like sort of replication that's being aborted somehow. But we were really quite surprised when we saw that. Again, we don't see this in everybody. We're now doing a lot of really sophisticated work to look at the local immune environment around this persistent virus. We're trying to sequence the virus. We're doing metagenomics to really understand what's going on here. And we've expanded this program and now have done probably 120 or 130 gut biopsies over the last few years in people with long COVID, as well as in people who have fully recovered, to see if we can see differences in signal between those two groups. So what we don't know is the form in which the virus persists. So we don't know if this is just fossilized, defunct RNA that is inert and not doing anything. We don't know if this is a non-productive infection where there's transcription and maybe translation, but active infectious virions are not being produced and there's no downstream infection. We don't know. And then we also don't know if this is active replication. And, like I said, the last set of data, like I showed you, might suggest that that could be happening. So we recently, I guess almost a year ago now, we put out sort of a position paper on this describing what could be happening. And if you're interested in this topic, I encourage you to check that out because it kind of lays out both the strengths of the data, but also the weaknesses and limitations of the data. So the question that stems from this is, does treating viral persistence matter? Maybe 3 years ago, there was a flurry of activity in the clinical space where drugs like Paxlovid and molnupiravir and monoclonals started to become available clinically, and there were case reports of people with long COVID getting access to these drugs and reporting improvement. And so that led to a lot of excitement that this might be a straightforward path to treat people with long COVID. Unfortunately, has not turned out to be straightforward. There are now 2 high-quality published studies of 2 weeks of Paxlovid, one at Stanford, one at Yale, that did not show a benefit. There's a 3rd study that took place at the Karolinska in Sweden that has been presented publicly that similar results to these. Recover Vital, the 900-person study that was done as the first Recover clinical trial, has not yet reported out. I expect that it will report out in the next 2 months, 2 to 3 months. But the data we have so far suggests that in all comers with long COVID, based entirely on symptomatology, there doesn't appear to be a benefit of a couple of weeks of antivirals. There are a number of reasons why that could be. That could be that, what I showed you in terms of active replication is not true. And these drugs would only work if there was active replication. So maybe these viral particles are still having an effect, but it's not treatable by this drug. We are still really struggling to identify people who actually have this for inclusion in clinical trials. You just can't screen people with gut biopsies and an analysis pipeline that takes months. It's just not feasible. And it could be that 2 weeks of treatment is just way too short. There are cat models of coronavirus persistence, and actually a paper that just came out in a veterinary journal like a month ago that suggests that you actually need months of treatment. I think it'd be very hard to tolerate some of these drugs for months. There's not safety data in people to allow us to try that right now, but it may be that just 2 weeks is not enough. We completed a study last year of a monoclonal antibody for long COVID called Outsmart LC. This was a funded by the Patient-Led Research Collaborative, with additional support from Polybio, and donation of drug from a company called Arium Therapeutics, which had been developing a monoclonal for clinical use that lost activity, but they were willing to allow us to use their drug as a sort of as a probe in this experimental medicine study to see whether we could identify any signal. So what we did in the study was we enrolled people with long COVID that was attributed to a variant against which this monoclonal was known to have activity. So basically ancestral, Delta, and early Omicron. It then lost activity. So everybody in this study had long COVID from an infection in 2020 through early 2022. And the rationale for that was it shouldn't matter what virus is circulating now. It should matter the variant that actually caused a person's long COVID. We did a number of baseline measurements, and then we randomized people to either get a single infusion of this monoclonal or placebo. And then we followed them for a year. And we measured basically everything. We did patient-reported outcomes for general health and then across a variety of sort of symptom groups. So physical function, cognitive function, autonomic function. We did objective testing related to those things. So, exercise capacity by 6-minute walk. We did neurocognitive testing using a computerized battery, active stand tests. We did a lot of biomarker work. And then people could actually opt in to doing things like gut biopsies and PET imaging. So it was quite a comprehensive study. The result was also, no difference between the groups in terms of the various outcomes we measured. This was quite disappointing. So we did not see a benefit in the overall cohort of the monoclonal compared to the placebo group. This was true across the summary scores, across the PROs in terms of specific symptoms, in terms of the objective testing. So quite disappointing, but I think an important finding. I've spent much of the last year thinking about why, so mainly I'll talk about that. So it could be that this monoclonal was just not a good monoclonal to study. It was the only one that we had access to. We have tried very hard to get access to other monoclonals since any monoclonal became available in 2020. And Arium, was really the only company willing to provide their drug. So it's possible that this was just the wrong drug. It could be that we did not enroll the right people. And as I mentioned, there was no clinically available, CLIA-validated measurement that was acceptable at the time that we enrolled this study. And so you need a clinically improved test to enroll someone into a clinical trial. It's a clinical decision, and that does not exist. So that was quite a challenge. It continues to be a challenge. We only gave people one dose. I mean, it's pretty unusual for one dose of anything to cure a chronic disease. And so it's possible that we really need more aggressive and sustained dosing. I don't think we would have been able to get that through the FDA at the time because they really sort of framed this as a safety study. It could be that this is the wrong strategy and you actually need combinations of drugs. So perhaps a combination of antiviral or monoclonal with a drug targeting the immune system. This is sort of a framework that is very familiar to me wearing my HIV cure hat. So in HIV cure, we don't think that targeting HIV alone is going to cure people of HIV. We always target the virus in combination with the immune system. And I've started to see success in the HIV cure space in doing that. So maybe that's an approach we need to be doing in long COVID. We are very interested in such an approach. And then, people always get angry when I say this. I mentioned people being really dogmatic on both sides, but it could be that this is just not what causes long COVID. I'm not ready to sort of throw this out yet. But it's possible that we are thinking about viral persistence as the most upstream cause, but there is actually something even more upstream of that. And maybe it's actually the immune dysfunction that is causing long COVID and happens to be causing residual fragments of virus to persist. So there are all sorts of models you can come up with. And I think that we cannot be married to any one model until it's proven. Um, the other thing that I'll show is I actually think that the most important finding from this study is the pharmacokinetic analysis that Amelia Deichmann in our group has done. And so what she has found is quite striking. So among this cohort of people with long COVID who either got drug or placebo, the single strongest predictor of whether somebody improved over the course of the study period was how high their baseline levels of their own antibodies against COVID was. I don't know why that would be. I don't know why someone who's had long COVID for 2 or 3 years and has a high starting antibody level would improve spontaneously over a 3-month observation period. It's hard to really explain that, but it was quite striking. Um, and those are objective measurements that an antibody level is an antibody level. Um, the other thing that she found was that if you look at the half of the cohort that had lower starting levels of antibodies, there was actually a dose response relationship with the drug in that subgroup. So people who had low levels of antibodies to begin with, um, and had higher levels of monoclonal circulating in their blood over the study period, so a higher area under the curve, were more likely to say that they got better. So I think that that's an important observation because I think we can learn from that to design a better study. And so I think that the next study of monoclonal should really try to target people who may have less strong immune responses to begin with. And I think the next study really needs to take a more aggressive dosing approach. So instead of a single infusion, I think we need to really maintain high levels of the monoclonals in people over, a 3 to 6 month period, to really give people enough time to respond. So that's important because positive development over the last year is this consortium called SPEAR, which you may have heard of. This was a group of us convened by Invivid, um, includes me, Amy Proal, who has done a lot of the really important work in sort of putting together the data for this, Akiko Iwasaki at Yale, David Petrino at Mount Sinai. And we're working with the team at Invivid, which is the company that makes pemivibart, which is approved in the US for prophylaxis, to design future monoclonal studies, either using that drug or sort of next-gen drugs. There's a lot of ongoing activity about this, and my hope is that this effort will be informed by the prior study we did with the Arium monoclonal. So I'll take 5 minutes now and just talk about where we're going with our clinical trials. As I mentioned, the types of clinical trials that we do at UCSF are really what we consider to be experimental medicine studies. So they're not, I don't run studies of 1,000 people trying to look for small clinical effects. The studies that we do are really focused on understanding the biology like we tried to do in the Arium study. So we've completed that study, and we recently completed a small study of using ensitrelvir as sort of a probe to understand whether using an antiviral can move anything biologically. We'll probably put the results of that out later this spring. It's not really focused on symptom improvement. It's really focused on the biology. We've also shifted beyond just targeting the virus. So I think one of the other really important results or really important outcomes from these negative trials is that it was really hard to convince the regulators, the IRB, et cetera, the grant reviewers, that it was worth doing studies that went beyond the virus until we knew that just targeting the virus in everybody is not going to solve the problem. And so that has given us more leeway to do more innovative studies, I think, targeting the immune system. So I'll mention two of these briefly, and then there's a lot of other stuff coming down the pipeline that we're really excited about. The most significant study I think that has started in the last year in the US is Reverse LC, uh, which I'm a part of. It's led by Wes Ely at Vanderbilt, Vince Marconi at Emory, Priscilla Shu at UCLA. It began as 4 sites, so Vanderbilt, Emory, UCSF, and University of Minnesota with Carolyn Bermonte up there. Um, and this is using baricitinib, which is a JAK inhibitor that is approved for acute COVID in people with severe illness. We actually have a lot of experience with in HIV in people with long COVID specifically who have neurocognitive symptoms. It'll enroll 550 people with neurocognitive symptoms, treat them for 6 months, and we'll measure many of the same things that we've measured in our other studies. So the primary outcome is actually performance on a neurocognitive battery. We're also looking at physical performance on cardiopulmonary exercise testing and then doing a lot of biomarker work. This study was initially funded by the National Institute on Aging, really framing long COVID as a type of cognitive impairment that can give us insight into like age-related cognitive impairment in people who often don't have all of the age-related comorbidities. Um, since the initial funding, it's actually been adopted by NIAID, and is now part of the Recover portfolio and is expanding from the initial 4 sites to 15 sites across the continental US. And so the goal of that is to enroll the study faster, have greater geographic diversity, and get an answer sooner. And so there are sites across the US that are opening like as we speak, which is very exciting. I should also mention Doug Fraser in Canada is doing a study that is using a different JAK inhibitor, upadacitinib. I can never pronounce it. Upadacitinib. So there are actually multiple JAK inhibitor studies happening now. There's also one I think that's happening in Boston. The other immune targeting study that we're doing that started in December is called Interrupt LC. This is funded in collaboration by the company Immunity Bio and Polybio, the foundation. It is actually a single arm study. Happy to talk about the rationale for that later. Of a drug called Anktiva, which is approved for bladder cancer in the US. And is a agonist of the cytokine IL-15, which is a regulator of T cell and NK cell function. Before COVID, I actually had a lot of interest in this molecule, again in HIV, as a way to improve coordination of the immune system in fighting HIV. And so it's basically the same rationale for long COVID that if maybe the reason that SARS-CoV-2 is persisting is that it's a failure of the immune system, and actually we need to enhance the immune system to clear residual virus. And so we are doing a study where we're enrolling people from our program in San Francisco, doing a variety of baseline measurements, including these tissue collections. And then we're giving them this drug, 2 doses, about 2 weeks apart. Study is very difficult to blind. People invariably get a large immunologic rash at the site of the injection. And so very difficult to do this as a blinded RCT. And so we decided to begin by doing an open label study to generate some safety data and some biological data. But this will enroll 20 people and is going well. And then, just in terms of what else is happening in the US, Recover Neuro, which is one of the other Recover trials, reported out in JAMA last month. That was of a cognitive rehab program. Unfortunately, that was a negative study. Recover Auto, which is focused on autonomic dysfunction, is using IVIG or ivabradine or both. Should report out this year. I'm actually not involved in that one, but it's a well-designed study, and I'm excited to see what it shows. Um, there's a study funded by the Department of Defense and led by a company called BioV of a drug called bezisterim. That study is called Address LC. That is happening in the US right now. This drug works on some of the same pathways as low-dose naltrexone. It's a TLR4 modulator. It gets across the blood-brain barrier. It also is an insulin sensitizer, so it's thought that it might improve glucose handling in the brain, which is something that other drugs of interest like GLP-1s may not do. Um, and so we're participating in that study. I was involved in helping to design it. That's going to enroll about 200 people at multiple sites across the US. We're screening for that now. A lot of interest in that. The next iteration of Recover TLC, in addition to the baricitinib trial, is going to be doing a trial of low-dose naltrexone in children, which is great because, as you know, there are very few trials in children. So this will be teenagers up to age 25. I am personally extremely enthusiastic about that study. There's going to be a study of GLP-1 drugs. I don't know the details yet. There's also a large study via Scripps that Julia Moore-Vogel is running and her team are running that is using GLP-1s, and that's being done remotely as a decentralized study. So a lot of excitement about that study. I don't know if they can enroll people in Canada, but that would be something to ask. And then Recover TLC is also going to be doing a trial of stellate ganglion block. Um, and then, there are a number of studies of things like rapamycin, and I mentioned the SPEAR group and next-gen monoclonal. So there's a lot of activity in the US happening on the clinical trials front, which I think is really encouraging. We're also very interested in prevention. COVID's still here. um, a lot of people got COVID this winter. There's all of this compelling data that suggests that what happens to someone during a current COVID infection will determine whether they have long COVID months later. We have data suggesting that simply the amount of virus that you recover from a person's nose in the first 2 weeks predicts whether they have long COVID 3 to 6 months later. Annie Antar at Hopkins has similar very interesting data. Data from the UK. Why are we not trying to prevent this if we don't have treatments? I just truly do not understand. Um, so we are really pushing trials of long COVID prevention. These trials would have been much easier to do 3 or 4 years ago than they will be to do now, but just because it's hard doesn't mean we shouldn't do them. And so what we really want to do are studies where we work with people who have a new infection, who don't currently have long COVID, and treat them for weeks during that infection and see whether we can move the needle on any of these post-acute things that we can measure, whether we can prevent long COVID. It's really a blind spot, I think, for the field and for the funders and for the government that we don't have these studies. And then the last thing, with which, if any of you have heard me talk in the last year and a half, is this program called Viper. So I think a big problem for the field is that there are great, well-meaning laboratories that do studies of a few dozen people with and without long COVID and see an interesting signal, biologically, and then make an announcement like, we have discovered the biomarker for long COVID. Whenever someone does that, my spider sense tingles because, that's not how diagnostic development works. And there's almost never any rigorous evaluation of these diagnostics. And I think it creates a lot of false hope, and people make a lot of promises that they can't follow up on, follow through on. And we're sitting here in 2026, and there's nothing I can offer to my patients in clinic, and nobody is happy about that. Like, we have been trying to, and not just us, raise this issue since we launched our clinical trials program, and it was not possible to screen people in. And that is a big barrier to industry engaging on clinical trials. And, it's just a terrible situation. And um, we can talk about the problem and talk about how it's hard to solve the problem and it costs a lot of resources to solve the problem, and this is why industry won't engage or whatever, or we can actually try to do something about it. And I think that we actually need to do something about it. It's a real shame that there's not a government sponsored diagnostics program in the US. Um, it's a real shame that there was actually a lot of resistance to this, I think, over the last few years from a variety of different groups, um, including sometimes patient groups. And there are effectively just way too few resources that have been put into this. And so we're trying to address this issue with this program called Viper, where we distribute in a blinded manner a gold standard sample set from people with and without long COVID to laboratories, commercial labs, academic labs, basic discovery labs, who are vetted and who want to try to develop a diagnostic. Um, and we take sort of the conflict of interest out of their hands, right? They study the samples in a blinded way. They provide the data back to a central analytic core, and we figure out what assays seem to be working, what assays are not working, what assays need to have, the ones that are not working great, how do we improve them? And the goal of this is to sort of like help everyone by eventually getting to a situation where we can say with confidence that, what the performance of different assays are and how we might use them in combination, and then provide guidance on how these could be further developed as commercial assays that can be used in a clinic. And so this is what I've been putting a lot of my energy into, because I think the lack of diagnostics is actually the single biggest barrier to progress in the field, and has been for 6 years and continues to be now. Um, and hopefully we can get this program off the ground soon. Okay, so just a couple of things to put on people's radar. The third long COVID international meeting was in Boston. Angela was there, a few others were there. Huge success. That was in November. The next one has already been announced. It'll be in France in November of this year. Also virtual, it'll be a hybrid meeting, but we're really pleased that this current iteration was a big success, and they've already announced the next one. And then the next Keystone meeting on long COVID and related illnesses will be in 2027. So I'm on the organizing committee for that meeting. There have been two of those so far in 2023 and 2025. Also a big success and a lot of excitement about that. I will end there with some acknowledgments. I want to especially acknowledge our program managers. Emily and Lynn really oversee the day-to-day of the program, our study volunteers, and many of whom started with blood but are now willing to undergo bone marrow collections, which is quite remarkable, I think, and our funders that make all the work possible. Okay, thanks so much for your attention. Happy to take questions.
That's terrific. Thank you so much. And terrific overview of what's going on in the US and elsewhere. And and cause for some hope in terms of future trials. So thank you very much. We have many questions and very little time here, so I am going to say that I will be selective. I'm going to pick questions from different people on different topics. And unfortunately, we really are unable to address personal health questions here. So I will ask, however, if and because we're not going to get through all of them, I don't know if Michael, you'll be able to have a look at the list of questions. We can ask, send them to you afterwards and perhaps post some of the responses
Sure.
later.
Yep.
Okay, that's terrific. Okay, so what do you think of the theory of spike antibody isotope switching from IgG1 to IgG4 in long COVID patients, enabling immune tolerance and thus viral spike persistence? Which treatments might help patients in this category?
Yeah, that's a great question. I think this is one of the like we need to be looking at all of these different pathways that could potentially be driving persistence or downstream of persistence or unrelated to persistence. I think um, we will see, I hope, a lot more studies targeting the immune response beyond just sort of generic inflammation, including I would like to see studies that target abnormal or pathogenic B cell responses, recycling or cycling of antibodies, um, and more to come on those therapeutics. I think in the next year, I hope.
Okay, thanks. Can an adverse reaction to IVIG, such as significant allergic or immunological reaction for someone that has long COVID, give any further clues as to the mechanism of immune dysregulation in long COVID sufferers?
That's a tough question. I don't know. We'd probably need to know more details about it. I mean, I think IVIG has its own, separate from COVID, set of adverse reactions that can happen. I've seen them myself in other indications. I don't actually have a lot of personal experience with IVIG for long COVID, but my position on this is that we can learn from everyone and everything, and so. Yeah, it's possible if we had the right information, right measurements.
Okay, thanks. If something persists, is long COVID truly binary? I mean, given it enough time and everyone will develop long COVID who once had COVID. Stated differently, I have seen people with immune dysfunction after COVID infection without showing long COVID symptoms. What would you, or would you characterize them as recovered?
Yeah, that's a great question. So that sort of gets back to that disturbing observation we made in our imaging program, where you know everyone, regardless of whether they had long COVID, seemed to have differences in their levels of immune system functioning. I think one of the most important questions for the field is like obviously we're very focused on long COVID because there are people who are incapacitated or debilitated like right now, right? But looking at the future horizon, what does all of this mean in terms of people who feel fine but may develop a comorbidity in 5 years or 10 years? Like it is, and is COVID either through viral persistence or another mechanism like immune dysfunction going to cause increased rates of cancer or cardiovascular disease in, my generation of, people in their 40s, right? We're not going to be able to answer those questions until enough time passes. But I think um, that question gets at the spirit of that issue. And yeah, I don't know the answer, but I think it is that should be something that is really important to study for everyone, for us academics, for clinicians, for epidemiologists, for industry partners who develop drugs to treat those things. We really need to have all hands on deck to figure that out.
Great, thanks. Does finding antigens or remnants of spike protein equal viral persistence? How does reality, I think, fit with that? So many catch COVID every 6 months on average.
Yeah, so that's a great question too. So these studies are so much more difficult to do now because we're all in a milieu of COVID at all times. The initial data that I showed, and I forgot to mention this, I normally do, was from an era where reinfections were exceedingly uncommon. And so we don't think that anybody in that analysis had an interim reinfection. Now it would be so much harder to do that study right now. But we need to figure out, there are ways to do it. The first part of the question though, so yeah, so this could be a reflection of reinfections, especially now. Um, you know, I use these terms kind of interchangeably, but if you wanted to add nuance, there's like true viral persistence, and then there could be viral antigen persistence without virus. And those may be different but related things. So that's a fair point. I think either of those things could be important and equally important.
Thank you. Angela, I see you've popped in. Do you want to ask a question, comment?
No, no, I just want to congratulate Michael on a very successful program that you have established, and thank you for all your work in this area. And also, thank you for a very thoughtful, clear presentation as well.
Yeah, thanks for having me. I'm happy to try to answer the list of questions if I can find some time. And thanks also for everything that you're doing up there. We're yeah, it's good to all feel like we're all on the same team, and this is a challenging time for us here. But hopefully we will continue to make progress together.
Still a relatively permeable border, so we'll hang on to that. But thank you so much. And we will send the list of questions. If you have 10 minutes here or there, that would be fantastic. Thank you,
Great.
everyone, for joining us. Please do have a look at the quick survey that we have. And we look forward to seeing you in February. Thank you. Thanks.
Okay, thanks for having me. Bye, everybody.
Bye-bye.
Thank you.